Today we will be covering chapter 15 from the syllabus and the text, which is titled Coordination and Response in Plants. Let me just put up the heading. And this is a short chapter, so we just might end up finishing a bit early today. Okay. So the first thing is sensitivity that we're going to talk about. So I'm not going to talk about it in detail, just what the concept of sensitivity is for a living organism. And so that is the ability to respond to a stimulus. Respond to a stimulus. And so everything we learned in the previous chapter was all about the sensitivity of the human, the biological sensitivity of the human body, responding to different stimuli and coordinating all those stimuli and then coming up with the appropriate response. Now, as far as plants are concerned, plants do not have a nervous system, right? So there's no nervous system and no hormonal system. But they are sensitive, they are able to respond to stimuli, right? Obviously, the range of response and the type of response is limited to other as compared to other organisms, but it is there, right? And one sort of group of chemicals or group of substances that plays a role in the sensitivity and coordination response for plants are called plant growth hormones, right? Now, that does not mean that plants have endocrine glands, right? There are no endocrine glands, there is no endocrine system. But the substance that are released are called hormones. So before we get there, what we need to talk is how this response comes about, right? And what we're going to talk about, right, is tropism. So tropism is the growth movements. Right, and they're related to directional stimuli. So we know that a plant usually grows up straight, right? So what's causing that upward growth? So there are two things, right? One is light and the other is gravity. Now in nature, light is coming from up above, right? So the source of light is above and the source of gravity is below, it's the earth, right? And so this direction of where the light is coming from has an effect on the way the plant grows. So what happens in plants are what are called phototropism, right? Photo meaning light, as we know from photosynthesis. And this is its response to the direction from which light is coming. So growth response to direction of light. And then you have gravitropism, coming from gravity, right? And that is obviously the growth response to gravity. And when you talk about response, right, the growth can be towards or away from either of these two. Now, is it random? Is it up to the plant's mood or something? No, right? It's pretty well documented and understood. So the plant is basically divided into two parts as far as it's concerned. You have the shoot, right, which is the stem and leaves. And roots. So as far as the shoot is concerned, it has positive phototropism, which means that the shoot, the stem of the plant, will grow towards the light. It will, right, grow upwards in nature, right? So it can be slightly different. We'll talk about that later. So that means it grows towards the light or upwards, and then it has negative gravitropism, right, which means it grows away from the earth. Right, because the gravity is coming from the earth is always downwards. The roots are positively gravitropic. Right, so they'll always grow downwards. Any questions so far?
No.
Okay. So moving on.
In a plant like gravitropism and phototropism can happen at the same time.
Gravitropism and phototropism happening it does happen at the same time.
Okay.
Right, so both of these tropisms are happening at the same time.
Okay.
Okay. So obviously, the positive tropism phototropism of shoots helps it to, you know, helps the plant position its leaves and flowers in the best, angle to get light. So the positive phototropism in the shoots is an advantage to the plant because it helps position the shoots, which means leaves, right, for maximum for maximum light exposure. And we know, right, sunlight again in nature is required for photosynthesis and all of that. Also, right, to make the flowers or have flowers available to pollinators. Right? And especially in the case of flying insects. Okay. And then, negative gravitropism allows it to grow vertically upwards and lift the entire plant up, and helps in competition. There are certain plants, right, that have rhizomes. Talk about rhizomes later. Rhizomes, and these are stems that grow underground. And an example of this is the common grass that you see. Right? If you ever pulled on grass and you see that it's just like a stem or a leaf that you can pluck out of the ground, it actually is pretty long, right? So if the leaf is over here and you pull on it, right, you would notice that the stem is running along, right, and you pull it up, and so you get this long stem coming out of the ground. That's a rhizome. Right, in that case, right, the stem is not negatively gravitropic. Right, so the general, right, it's a general rule that stems are positively phototropic and negatively gravitropic, but then there are exceptions everywhere, right? And so this is one of them. But right from the stems, the shoots, so the leaves and all, right, they are negatively gravitropic. Okay. Now, with that, right, what is the sort of chemistry or what's going on inside the plant that is causing this gravitropism, right? And that is where the plant growth substances come in. And the one that we're interested in, there are many, but the one we're interested in is called auxin. So plant growth substances. right? And as I mentioned, what we're interested in talking about is auxin, AUXIN. And this thing is produced in the tips of actively growing roots and shoots. Right? And so I'll share the illustration for this, but right, what happens is it's produced in those regions, right, actively growing regions. So basically the tips of the roots and the shoots, which are growing and along, right? Uh, Actively growing meaning that there is active cell division going on, right? And then what happens? So I need to go to the next page, right? From the tips,
Can you explain it again?
I explain which part.
The auxin part.
Yeah, so right, auxin is a plant growth substance. It is produced in the tips of roots and shoots where active cell division is occurring, right? And after it is produced from the tips, it diffuses to the region below the tips, right, where you have new cells. Right, and it affects cell elongation. Okay, now how does it affect cell elongation? It can either promote cell elongation or it can prevent it, and we'll talk about both the scenarios. So what happens in shoots? Right, first of all, it depends on the direction where the light is coming from. So if light, so if this is a shoot and light is coming from directly above, so what happens is that, let me just draw this a bit bigger. Right, and you've got light coming in from above, and so this is the region where active cell, right, division is taking place. And so the auxin is also produced here. And from there it diffuses to the region below and promotes cell elongation right, uniformly, let's say. But if there's a different case, and you may have noticed sometimes plants are sort of growing in like one direction, they're not going up straight, right? You may have noticed that plants are growing like this. And so that really, one reason for that is where the light is coming from. And so let's say the light is coming in from the side this time. And so same thing, right? You have the area of active cell division. And this time what happens is that the oxygen diffuses to the region in the shades, right? So this is the shaded region where there isn't enough light, let's say, or no light, right? And the oxygen accumulates here. And so what that does is it promotes cell elongation in shaded region only, right? Because that's where it's accumulated, right? That's where it's diffusing, right? By the way, oxygen travels by diffusion. So, right, if you're asked in an exam, right, how did oxygen move from the tips to the elongation zone? And so that is by diffusion. So because it promotes a elongation, it's in response to the light, right? It accumulated in the shaded region and it promotes elongation only in the shaded region. So what's happening now is that the cells of the shaded region become longer, as compared to, obviously not the best illustration, right? The cells in the shaded region become longer. So over here you have more elongation, right? Over here you've got less elongation because you didn't have a lot of oxygen going over there. And that causes the stem to bend towards the light, right? The light was coming in from this side, remember? So that's what happens in stems. Does this make sense?
Yeah.
Okay, that's good to know. So, right, that is how you get,
Uh,
yeah,
Oxygen is like present in every part.
yes. There are other growth hormones as well, right, in plants. And perhaps not all of them may be present in all plants, but right, oxygen is also like, uh, there are different types, but there's like they're generally called oxygen. So right now we're not discussing the different types. We're just talking about
Okay.
generally this kind of growth substance that brings about such an effect is called an oxygen.
Okay.
Okay. So that's right. So that is why we have positive phototropism in shoots. It is because of the oxygen's response to light. Phototropism. Now, as for the roots, right, since you understand the concept, what happens is, right, you have this root. I'm going to draw it horizontally, right? And obviously you have the earth over here. And so it has positive gravitropism. Just a minute. Right. So what happens is, right, auxin is produced and it accumulates on the side that is right closer to the earth. So in this case, the lower side, right? So auxin accumulates. I'm saying lower, depend like based on the diagram, right? Basically, it accumulates at the side where which is facing gravity, so to speak. And in this case, what it does is it auxin inhibits cell elongation. So it prevents cell elongation. We talk about it. It can have two effects.
Yeah.
So in the root, what it's doing is it's preventing cell elongation in the side where it is accumulating. So it's accumulating on the lower side, right? It's preventing cell elongation. So the upper side of the root has more cell elongation taking place. And so what we see is that the root will bend downwards. Now, obviously, right, if a root is growing straight, right, then similarly to this scenario, right, it will keep growing straight down because the effect of gravity is the same throughout, right? It's only when it's at an angle that, right, so you've got more elongation over here. Due to auxin not being present here. Again, auxin is on the lower side, right? So there's auxin is over here, right? And you have more elongation on the upper side of the root. And that's how you get positive gravitropism in the roots. Clear?
Yes.
Okay. So the mechanism by which cell elongation occurs, right? There are many things that happen, but right, basically, the cells take in more water, among other things. The vacuole of the cells, so these are plant cells and they've got a large central vacuole. They expand, right? They become larger, and that causes an increase in cell size. Right? And thereby you get elongation. Now I'll take you to the book and show you some illustrations and go over an experiment, right? And the experiment is important to understand the concept and also with regards to examination questions, right? Can you see that? Yes?
Yes.
Right? So the auxin producing region is actually the very tip, right? So I drew it a bit more, right? It's actually the very tip. That's where cell division is occurring. The auxin is transported down, right? And so you have, right, vacuoles in the cytoplasm of the cell, and then they take up water right? And they become larger. So vacuoles join up to form a central vacuole which absorbs water and expands cell lengthwise by increase in turgor. Right. So as far as the illustration to understand this, obviously follow the one that's given in the book.
So like auxins are present in every part of the leaf.
Not the leaf, the stem.
Okay.
Right. The
So
shoot.
like
The shoot is the stem and the leaf. So auxins are present in the tip of the shoot.
okay.
Right. Not the leaves themselves.
They are like transported into the leaf then.
Not the leaf. Right. The position of the leaf is also adjusted by the plant depending on where the light is coming from. Right. But it's about the direction in which the shoot of the plant is going. Does that make sense?
Yes.
Okay. Right. So again, right, you got light coming in from here. It goes evenly. It goes up. If light is coming in from an angle, there's more on the shaded region, and so right, the elongation is more on the shaded region, and so it bends towards the light. As for a root, right, it accumulates on the side that is towards the gravity, right, towards the earth, and it inhibits, and there's more elongation on the upper side, and so you get it growing towards the earth. So that's positive gravitropism. Now the experiment, what they did was they took the uh, seedlings, right, germinating seeds from a monopod plant such as wheat or grass, and the shoot is covered by a protective sheath called a coleoptile. What that coleoptile does is it prevents damage to the new leaves as they push through the soil, right? And it also shows response to light and gravity, just like we studied, right? So it has positive phototropism and negative geotropism, or not geo but gravitropism, right? So what they did, because it's also the growing part of the shoot, right? So it's this part. They cut it off. In like they had 4 experiments. The first one that in the first one the tip was removed, right? So they cut it off. The second one they placed it whole as it is. In the third one, right, they covered the coleoptile with black paper. And in the fourth one, what they did is first they cut it off the tip and they placed it on an agar block for 6 hours in the dark, right? And then the agar block, right, first they sort of let it be exposed to light, right? And then after that, they place the agar block on top of the cut shoot, right? So the agar block is transferred to the coleoptile, the tip from of which has been removed. So these are the 4 scenarios. So because it's the growing tip, right, that's where the auxin is produced. So if the tip is cut off and you can see light is coming in from one direction, right, in this case the left side. So what they noticed over here in A, where the tip has been cut off, that no elongation occurred at all and no bending. So neither was there any elongation nor was there any bending. So there was no change observed, right? So let's do this one by one. What happens in A? Because the source of the auxin has been removed, it is not responding to the light, right? Auxin is needed to stimulate elongation and a response to light. It could also be argued that the tip provides cells for growth, and this source of cells has been removed, right? So because they're not getting any auxin from the tip, right, you're not seeing any response to the light. In the second scenario, where it's just the growing shoot with the coleoptile, the tip and everything, right, you see it bending towards the light and it's elongating, it's growing taller, right? So it's moving towards the left. It's growing taller, it's moving towards the left. And that makes sense, right? According to what we studied. Auxin is produced by the tip, it diffuses down the coleoptile and collects in the shaded side, right? Cells of the shaded side respond and elongate faster than on the light side, causing it to grow towards the left. That's exactly what we would expect. In the third experiment, or the third setup, do you have a question?
No.
Okay. So in the third setup, they covered it with black paper. Now, what you would expect is that it's not going to be affected by the light, but auxin is being produced. It's just not going to be affected by the light. And so it should grow straight up, right? Because this auxin is there, right? It's just not responding to the light. And that's exactly what happens. Auxin is produced by the tip and diffuses down, causing all cells on both sides to elongate at equal rate. So it's uniform, right? And it causes an increase in length, but it's not bending because the black paper prevents the light from influencing the auxin. And so there's no response to the direction of light. So you get elongation, you don't get any bending because, right, it's covered with black paper, so it's not affected by the light. All right. Now, D is interesting, right? What would you expect to happen in D? So even though they cut off the tip from where auxin is produced and diffused to the lower parts of the growing shoot, they placed it on the agar, and then they placed the agar block on the cut shoot, and then you observe a response similar to B. The coleoptile grows taller and bends towards the light. Now, why is that happening? That's happening because of diffusion, right? What does that mean? The auxin that is produced in the tip diffuses into the agar block, right? It was left on the agar block for 6 hours, right? And so whatever was there, instead of going into the shoot because it's cut off, it can't do that. It goes into the agar block, right? And so when the agar block is replaced on the cut stem, the cut coleoptile, the auxin diffuses from the block into the rest of the stem, and then it accumulates in the shaded region in response to light. And so the shaded region responds by having more elongation, and so it bends towards light. So not only does it grow, right, it bends towards the light. So this also tells us that it's not necessary to have the like, let me rephrase that you can sort of stimulate this response by artificially applying the plant growth substance to the plant, and this has been done, right? So it mentions over here that chemicals, this is going further, right, but still taking D, right, chemicals can be manufactured which are very similar to natural growth substances and may be used to control various stages of growth and development, right? So the auxin in the agar block is not something you would find in nature, obviously, right? But you could think of it, even though it did come from the cut part, which is natural, the agar block itself is not natural, right? So if you have a stem that is cut, and forget the agar block, right? You got the synthetic growth substance, and you apply it to the tip, right? So just taking a look at the illustration, instead of the agar block, you apply a synthetic growth substance to the tip. You will observe the same response as a normal shoot, because the point is that the auxin is what produces the elongation and the bending, right? And as long as there's auxin present, even if it's not coming from the tip, it's artificial, or it's coming from the agar block, you will notice a response, right? Does that make sense?
Yes.
Okay, and that's all we need to know.
So it's the end.
Huh?
So it's the end of the chapter.
It is the end of the chapter, yes. Right? Now, the next chapters are development and all of that. I think instead of jumping into this, I'll go back and do chapter 2 that I left, and then with that, right? So I left chapter 2, which is classification, so we can do that. And with classification, we can then do chapter 19, relationship of organisms with one another and with the environment, right? So we'll do
Okay.
chapter 2 and then 19 with the time we have left. And then after that, we can do 16, 17, and 18.
Can we do the biotechnology first
Uh-huh.
instead of classification?
Classification is what we'll start tomorrow.
Uh, I was saying that we can start from like biotechnology
Biotechnology,
instead of classification.
biotechnology first.
Yeah.
We could, we could do that, but yeah, okay, I'll see if we do
Okay.
that first. It's
Thank you.
it again. This is also not a long chapter, so
Yeah.
maybe not in one class, but two classes definitely. There's a lot of reading in this, right? So what I'll do is I'll cover the basic concepts of whatever there is, and then I'll leave the reading to you, the stuff that you can just read and figure out. Okay.
Yeah.
So I'll leave that to you, and I'll just cover the basic concepts of this chapter. All right.
Okay.
All right. All right. See you next time.
Last.
Allah Hafiz.